సార్ గుడ్ మార్నింగ్ సార్. కృష్ణానంద్ కులకర్ణి గారు. హలో.
Good morning, sir.
సార్ గుడ్ మార్నింగ్, గుడ్ మార్నింగ్. వెల్కమ్ టు డాక్టర్ వైఎస్ఆర్ హార్టికల్చర్ యూనివర్సిటీ.
Thank you, sir. Thank you.
సో పీపుల్ ఆర్ జాయినింగ్. We will wait up to for 10 minutes.
Okay, sir. Sure.
మేడం అక్కడ తీసుకురామ్మా ఇది డబ్బులు పెట్టుకుని వెళ్ళేటప్పుడు తీసుకుని కోసం అదొకటి అన్ని
Sir, good morning, sir.
Good morning, madam.
Sir.
గుడ్ మార్నింగ్ ధనంజయరావు గారు. కల్పేసి
సార్ గుడ్ మార్నింగ్ సార్.
అదే విసి గారు మనల్ని కంటిన్యూ చేయమన్నారు.
అలాగే సార్.
ఓకే
5 minutesలో స్టార్ట్ చేద్దాం సార్.
ఓకే ఓకే
రవీంద్ర సార్ రాలేదు. మేడము రవీంద్ర సార్ టూర్. దమ్ము 23-24 ఇది అబ్జర్వేషన్ బుక్.
101, sir.
Good morning, all of you. Welcome to Dr. YSR Horticulture University, Venkatramanagudam, West Godavari District, Andhra Pradesh, for the 21st Global Connect series in collaboration with the Delaware State University, USA, on deciphering the genetic factors regulating heat stress tolerance in blueberry. And respected Honorable Vice Chancellor, Dr. K. Gopal Garu, with his visionary leadership, we are conducting 21st Global Connect series. Thank you very much, sir, for giving permission to conduct 21st Global Connect series. And today, guest speaker, Dr. Krishnanand P. Kulakarni, Postdoctoral Research Associate, Department of Agricultural and Natural Resources, Delaware State University, Dover, USA. Welcome, sir. Welcome and good morning to Dr. Krishnanand P. Kulakarni.
Thank you very much, sir. Thank you.
Sir, all university officers, Dean of Horticulture, Dr. Naram Naidu Garu, Dean of PG Studies, Dr. Venkatramana Garu, Director of Research, Dr. Madhavi Garu, Dean of Student Affairs, Dr. Suryakumar Garu, University Librarian, Dr. Ramanandam Garu, Controller of Examinations, Dr. Prasanna Kumar Garu, Director of Extension, Dr. Govinda Rajal Garu, Registrar, Dr. B. Srinivasulu Garu, Controller, Dr. Srinivas Subramanian Garu, Estate Officer Ramana Garu, and Director P&M Cell, Dr. Vijay Padma Garu, all Associate Deans of Colleges of Horticulture, and all principals of polytechnic colleges, all heads of research stations and KVKs, and all teachers, scientists, and students of Dr. YSR Horticulture University. Welcome, sir. Welcome to today's 21st Global Connect series. And the rapporteurs, Dr. C.H. Nalini, Assistant Professor Entomology, College of Horticulture, VR Gudam, and Dr. S.B. Ramya Lakshmi, Assistant Professor, Agriculture Economics, College of Horticulture, Anantaraspeta, will act as rapporteurs for this program. And I invite that guest speaker. Before going to invite our guest speaker today, just I will give the brief biodata of Dr. Krishnanand P. Kulkarni garu. Sir is presently working as a postdoctoral research associate, Department of Agriculture and Natural Resources, Delaware State University, Dover, USA. And sir studied MSc in microbiology, Swami Ramananda Tirtha, Marathwada University, Nanded, Maharashtra, India, and a PhD in biotechnology, National Institute of Plant Biology, IRI, New Delhi, and SRTM University, Nanded University, India. And sir did a PhD under the guidance of Dr. Mahopatra, Chairperson, Protection of Plant Varieties and Farmers' Rights Authority, Ministry of Agriculture, Government of India, and a Farmer Secretary, DARE and DG of ICIA. During his doctoral research, he developed the dwarf and high tailoring mutant of rice. For that, he used QTL mapping, positioning cloning, microarrays, and PRT, PCR approaches for characterize of dwarf and high tailoring mutants of rice. And his current research at Delaware State University is working on genetics and breeding for horticulture crops, primarily blueberry, maple, and strawberry. In blueberry, he used different approaches, GWAS and transcript analysis to study the heat stress responses and validate gene through real-time PCR and KSP assays. And he's investigating evolutionary relationships to the understanding the origin of domesticated blueberries. And his goal is genetically enhanced heat tolerance of popular blueberry cultivars. And previous postdoctoral research, Kianchuk National Institute, he worked in Republic of Korea, and a project genetic dissection of domestication and forage-related traits in soybeans, and Oklahoma State University, USA, and he worked on the project functional genomics of leaf architecture and seed development in chickpea. And presently investigator in this project, and he's handling 6 million US dollars project on blueberry. And he authored 36 peer-reviewed publications, 2 book chapters, 10 oral talks, and 20 posters, abstract presentations at national and international conferences, workshops. And so under his guidance, 10 graduate and undergraduate students are working. He is guiding and guided, and he is guiding presently. And sir is having 2 children at Maharashtra, and presently working at Delaware State University. This is the brief biodata, sir. Welcome to this Dr. YSR Horticulture University Global Connect series. And sir, kindly proceed. Thank you, sir.
Thank you. Thank you. And thank you all other dignitaries. So let me share my presentation.
Yes, sir.
See my presentation?
Yes, sir. Audible and visible. Proceed.
Thank you very much. So, as mentioned by Dr. Rao, I am working at the Delhi State University. My name is Krishnant Kulkarni, and then I work primarily in blueberries, along with other horticulture crops such as strawberries and maple. So today I will be presenting our work, which is on deciphering the genetic factors regulating heat stress tolerance in blueberries. So blueberries, it is considered as one of the superfoods, especially it is very important crop for the US. For India also, there are some farms which are now cultivating blueberries, but it is a very new crop for India. But for the US, it is a very high value crop. Blueberry belongs to Vaccinium section, Cyanococcus. It is a perennial shrub which is cultivated for its edible fruit. In the world, United States is the largest producer of blueberries, producing, for example, 3.259 in 2023. So this was the production of blueberries. Apart from USA, Canada is also a major producer of blueberries, but also even being the largest producer of blueberries, US is also largest importer of blueberries, and they are mostly imported from neighboring countries like Peru, Mexico, and Chile. And if you see, this graph here, the US production.
Sir, you close another laptop or anything. Two are
No, I don't
working
have.
at our
I don't.
place.
I have
Okay.
only one laptop. I think
Okay.
someone else.
Yeah. Okay,
Okay, so if
continue.
you see US total imports, the production is relatively still increasing, but overall stagnant compared to the demand. So therefore, the production of blueberries in the US also needs to be increased to meet the demand. So blueberries are considered superfood because of its health benefits. It is a rich source of phytocompounds such as anthocyanins, phenolic acid, flavonoids, and tannins. Even a bowl of blueberries, like in the daily diet, it can give you a super source of antioxidants. It can boost immunity. It can reduce the risk of heart disease. It can boost the brain health and then regulate blood sugar or reduce inflammation. It also promotes healthy skin, and it also aids in preventing dementia. So they have a lot of health benefits, and they are proven as given in these papers. So because of which the demand is ever increasing for blueberries. So if you, this is a little bit about the introduction of blueberry types to just to understand what blueberries and what types. So mostly they in the US, they are considered as a northern species and southern species. Northern species means those which are grown in northeastern part of the US and Canada, where there are very high, like very cold temperatures. So those blueberries, they require high chilling house. So in blueberry or in such a berry, horticulture berries, they require chilling hours, means minimum of like 500 or 1,000 hours of temperature less than 7 degrees. So that those are called chilling hours. So the northern species, they require high chilling hours, but they have large fruit size. But the southern species, which are grown in the southern side of the US, like Texas or Florida, Georgia, they require less chilling hours, and then they grow in warmer climates. But relatively, they have small fruit size. The species wise, the Vaccinium corymbosum, it is a high bush. This is the major cultivars, like most of the popular blueberry cultivars, they belong to Vaccinium corymbosum species. And The other species like Vaccinium virgatum,
ఇంగ్లీష్ మాట్లాడుతున్నాడు. యా మరి ఈయన ఆడబట్టుకున్నారు తమ్ముడు పేరేమో
so these rabbiti, these are also very large size fruits, and they can grow in relatively warmer temperatures like Texas or something, and especially like in India also, the cultivations of rabbiti, they are now growing. But if you see this right side of this, these are all wild species like Vaccinium boreali or Dharavi. So they have relatively small fruit size, and then they are grown mostly in like rocky uplands and open conifer forest, and they are very relatively bitter in taste also. So they are not favored in taste, but they grow at warmer temperatures, and that is why they form an important part of breeding in blueberries. So for the growth requirements, blueberry, it is a shallow rooted plant. It requires a soil that has holds moisture, but it also drains well and it doesn't stay wet. For example, like a sandy soil or peat moss, they can drain the water easily. So in such soils, blueberries grow well. Also, the soil must be acidic, and the non-acidic soil, it will stunt its growth. The pH of the soil, it must be around like 3.5 to 5. If it is more than that, then you will not have better yields. So this is about like general information like growing blueberries, like what location they like, or at what when to plant, and then fertilizing, then pruning. Pruning is very, very important for blueberries because it is like it's not an annual crop, it's a perennial crop. And then the harvesting season here, it can be from late May to mid-August. But if you compare this to in India, it will be somewhere like from August to November if you are growing in some of the regions, because it requires cooler temperatures. Heat stress impacts plant growth and productivity. So this is a very, very hot topic recently because of climate change. If you see this graph, it's the average number of heat waves. They are continuously increasing, and also the duration of the heat waves every decade. So it is also increasing. And the right side, this is the recent global climate report of the National Center for Environmental Information. And it shows that the average global surface temperature ranked highest for the February 24 since 1850. So it is very, very essential to have the plant varieties which can sustain these heat waves, not only the temperature, but the inconsistency of those heat waves as well. So this is one of the figure from a recent review in Plant Genome. It shows that the heat stress affect at all stages of the plant, but particularly the photosynthesis plant, which immediately, as soon as the plants are subjected to heat stress, the photosynthesis is affected. In blueberry, heat stress can cause lot of damage, especially to the fruit, because it can cause the desiccation and then the coloration. It can also affect the overall quality. So therefore, it is very, very essential that the blueberries, they are resistant to such a heat stress. So how can we provide that resistance? It is the exploiting wild blueberries. So as I said earlier, the southern species, they are better adapted to warmer climates, although they don't have better taste and size, but they definitely have the genes and alleles. Those genes that can give the plant ability to survive under warmer warmer conditions. So the blueberry is recently domesticated, just before 100 years. So during these 100 years, what researchers have done, they have crossed these northern species, which require high chilling hours, with the southern species. They require low chilling hours, and then they have come up with varieties like rabbit eye. So our objective was also to identify the QTLs and genes which are related to heat tolerance in blueberries. So for this, to accomplish this objective, a scientist, Dr. Nikoli Vorsa from Rutgers University, he developed a mapping population. So this Dharavi, this is a southern species, and Corimbosum, it is a northern species. So he made two different crosses for between the southern and northern species, and because blueberry, it is a cross-pollinated, so you require two different F1s to make a pseudo F1 population. So this population, it has around 1,300 plants, but for our research we used only 260 plants from this population. So we phenotyped these plants for heat stress tolerance. We used 40 degrees centigrade temperature for 4 days in growth chamber conditions. And after 4 days of exposure, we measured the quantum yield and OJIP, that is the rapid fluorescence transient assay. So the OJIP assay, it provides the current state of photosystem second inside the cell. And along with this, we also measured the vegetation indices, which are like normalized difference vegetation index, NDVI, then simple ratio, and then green nets. So these two handheld devices, they were used to measure the traits. For the measurements, we use like 3 random leaves, and then for the fluoropane, we need dark adaptation because all the photosystems in the chloroplast, they will be in the oxidized and they will be open for light absorption. So once the light beam is illuminated, the chloroplast begin absorbing light, and once the the chloroplast they absorb the light, the remaining light which is not absorbed, it is measured by the instrument and it is recomputed as a quantum yield. So that gives the current state of the photosystem 2, which is very, very sensitive for heat stress. So we conducted these experiments for 2 years, and then the data was combined and it was used as a phenotype data in GWAS. So if you see this figure, the tolerant plant, they did not have much of the damage to their leaves. The sensitive plants, they have the scorch. So we measured these leaf scorch on 0 to 5 scale, 0 being like tolerant and 5 means like completely sensitive. And then this leaf scorch score, it was positively correlated with the chlorophyll fluorescence parameters such as quantum yield. So these are the box plot which shows the variation for different chlorophyll fluorescence parameters. So these 10 parameters we used for GWAS analysis. So this is the frequency distribution of those parameters, and if you see, it is a quantitative distribution of all the traits form, and then the distribution, it is also transgressive segregation for most of the traits like F0 like this. So this shows that these traits, they are amenable for QTN analysis or GWAS. So for genotyping, we use the GBS assay, which we have already shown that it is able to produce a large number of SNPs in blueberries. So using these SNPs, we map those reads with Draper genome sequence, which was sequenced in 2019. And then this GBS, we had almost 976 million raw reads. Average number of reads, it was around 3.6 million. And then around about 84% of those reads, they were mapped to the Draper genome sequence. And the distribution of the SNPs across the 12 chromosomes, it was even, and then almost all the regions, they had the SNPs. So this shows that the GBS, it was indeed giving a lot of SNPs for all the regions. So in this analysis, after filtering, we used 126,000 SNPs for our GWAS analysis, which was done using a multi-locus mixed linear model. And then the SNPs underwent a false discovery rate for the significance. And then through this analysis, we identified 1,323 SNPs, which are significantly associated for all these parameters which in the high temperature conditions. And these SNPs definitely can be important in differentiating the plants, segregating for the respective trait. So these are the Manhattan plots showing the association of SNP with the measured parameters. So for further analysis, we choose those SNPs which were associated with more than one chlorophyll parameter. So that way we shortlisted those parameters. There's SNPs up to 281. And this is the distribution of those SNPs. About 179 SNPs, they were present in the genic regions, whereas the remaining were intergenic. The chromosome 1 had the largest number of SNPs. So when we did the BLAST analysis, we found these are the important genes which may be involved in heat stress tolerance in blueberries. The genes like a chaperone protein or the Wernicke transcription factors, they are well-known genes involved in abiotic stress, including heat stress. So along with this, we also did the morphophysiological analysis of this plant. So this is at the left, this Corymbosum. This is a northern species, whereas Daru is the southern species. So if you see the plant morphology itself, the Corymbosum, it has broader leaves, and the orientation is perpendicular, whereas the Daruvi, it has clustered leaves. They are oriented in multiple directions. The leaves also have wax coating, and their size is also very small. That helps to prevent water loss during the high temperature. So morphologically also, these plants, they got the benefit to sustain the higher temperatures. So these are the laser scanning confocal imaging of the stomata. So when the temperature rises significantly, plants always tend to close their stomata to minimize the water loss. And if you see here, in Daruvi, the stomata, they were like they were reducing, the size was reducing, which shows that they were closing the stomata when the 9-hour stress was given. But in terms of Corymbosum, such a closure was not observed. So this shows that both the species, they have differential response to high temperature. So these two species, we give the heat stress for 6 and 9 hours, and then we did the transcriptome analysis. So this was the transcriptome analysis we did, and then the reads were mapped to again the same genome, Draper genome sequence, and then we identified the differentially expressed genes which we published in IJMS. So we found around 325 common genes which are differentially expressed for all the 4 data points, like foreign bosom as well as Dharavi for 6 hours and 9 hours. So these genes we selected, and then they were validated using the QRT-PCR. So we selected 6 genes for that, and then the expression was similar to what we observed in the RNA-seq analysis. So these genes we compared with the GWAS results. So we wanted to integrate the GWAS analysis with the transcriptome and then identify the common genes. So in this is the pathway analysis. We found major pathways like metabolic pathways, and then biosynthesis of amino acids and plant pathogen interactions. So these were the major pathways in which these genes were expressed. So when we compared those transcriptome GWAS, we found 11 genes. I think my equation. Yeah, so when we compared the GWAS with transcriptome data, we found 11 genes which were common in between the GWAS and transcriptome. And there were common genes, well-known genes, for example, heat shock proteins and then NAC domains. These are well-known genes expressed are responsible for heat stress tolerance. Apart from that, we also found a gene like a 3-ketoacyl-CoA synthase 6. This gene it has been shown to enhance the drought tolerance by regulating wax biosynthesis. So this gene also is very, very important for abiotic stress tolerance. And then the other gene like the transcriptome termination factor. So this gene also, it is required, and it has been shown that their disruption confers heat tolerance in Arabidopsis. So we feel that these genes are very important for imparting heat tolerance in blueberries. So for that, we are now developing caspase for these SNPs, and then after validating, we plan to use them in introgression and breeding. So to summarize our study, in this study we phenotyped about 266 plants for heat stress traits, and then most of these traits they showed a quantitative distribution, and some of them they had a transgressive segregation, and therefore we performed a GWAS to identify the significant SNPs. Validation of those SNPs using the CASP assays, it is in process. Parallelly, we also did the transcriptome analysis, which identified about 325 DEGs, and then they were also validated using QRT-PCR. And when we integrate these two analyses like GWAS with the transcriptome, we came up with 11 candidate genes. They were involved for heat stress tolerance in blueberries. So we feel that these results will provide insight into the mechanisms underpinning the response to high temperature or heat stress in blueberries. So these are some of our papers recently published. Those who want to have more information about our work, they can refer to these papers. So this is our group. Dr. Kalpata Melme, she is my PI, and I thank her for giving me the chance to carry out this work. And Dr. Satya as well, he is a professor here in the Delaware State University. Under both of their guidance, we carry out this research. Dr. Nick Warsa, I also thank Dr. Nicole Warsa, who is a breeder at the Rutgers University. Most of the plants they provide the plants for our study, and then we make those crosses at our center. And then the genomics support was provided by Dr. Umesh Reddy sir, so I thank him for the GBS and analysis. And then Dr. Massimo, who is a scientist at the North Carolina State University, he provided those reference genome sequences for which we mapped our reads. And then we thank funding support from the USDA awards. Yeah, so thank you very much. So if you have questions, I will definitely take.
Thank you, sir. Thank you for your excellent presentation on genes regulating heat stress and tolerance in blueberries. And you have told that two types of berries are there, that high chilling requirement and low chilling requirement varieties and crosses, and 325 genes are responsible, and studied out of which 11 genes are responsible for the heat tolerance. Thank you very much for your presentation on blueberries heat tolerance. Now it is open for the discussion. Any question and answers from the
Thank you.
university officers or any teaching faculty, students?
Good morning.
Good morning.
Dr. Krishnanand, good morning. I'm Dr. Srinivasu, Registrar.
Yeah, thank you, sir. Thank you very much.
Yes. So this is blueberry. Yes, as you said, this is one of the important commercial crop which is being grown in America. Now it is also a new crop to India. Nowadays, a lot of attention is being paid to this crop because of its numerous health benefits, particularly because of its high contents of antioxidants, flavonoids, and all, particularly for the benefit of the cancer patients.
Yes, sir.
So are there any popular varieties grown there with some tolerance to heat and high temperatures?
Yes, sir. There, as I said, like if you wanted to grow really in cold temperatures, then there are very good varieties like Blue Crop or Blue Jay, Chandler. So those are very good varieties for the taste as well as size. But if you wanted to grow like our temperatures, like India, so then you need either Southern High Bush or Rabbit Eye cultivars. So for that, we already have like a Legacy. So Legacy is very good Southern High Bush cultivar. Then we also have Jewel. So Jewel is also good. Biloxi is also good. So those are, and then Sharp Blue is also good. So yeah, there are several varieties. Mostly if you consider Rabbit Eyes, so and then Southern High Bush. So they are good for our temperatures. So especially like in Shimla or those kind of temperatures, it will be good.
In northern India, there is a possibility of growing these blueberries. As you mentioned, some of the varieties are maybe they may be suitable for this northern part of India. And in southern part also, there are some areas like Ooty, Kunur and area where the temperatures also maybe the chilling hours, whatever you told that 500 hours of chilling, those will be met particularly during October to January, February months.
Right. But the Rabbit Eye, they don't require up to 500, so even 200, 300 is enough.
Yes. So with that, if it is 200 hours of chilling temperatures, no. I think we can try for some of these varieties in India also, because there is a lot of demand for these blueberries nowadays.
Yeah, definitely, sir. And then there are already some farmers and they are really
Yes.
doing, which I read several reports, and they are growing in Northeast also some parts, and then in Ooty also, I think.
Okay. Good. And other factor is, as you told, that it requires highly acidic soils.
Yes. So that can be managed. What can be done is like you can use first the peat moss, which is like it can easily drain.
Okay.
And if you don't have that one, then the sandy soils can be used. So you can mix perlite, sandy soils along with our soil, right? Or like, and then you can use like vinegar or that when you watering, mix with vinegar so that you provide the acidic conditions. So that should be good.
Thank you. Thank you, Krishnanand.
Anybody? Thank you, sir. Anybody from teaching faculty, students? Any students, any scientists?
Even this is, sir, just as information, compared to strawberries, this is a very healthy fruit. Like the strawberries, is it attracts a lot of pests, but in blueberry you don't have that concern. So that way
Yes.
also very useful.
Then how about this? When you compare strawberry and this blueberries, the shelf life for the fruit, how many days it will be there for without any damaging, I mean, the quality aspect? How many
Yes,
days we can keep it under normal conditions?
yeah, normal conditions you can keep up to like 2 weeks, 3 weeks, then nothing happens, sir.
Okay, okay. In 3 weeks
But
time.
it has otherwise like a strawberry, it immediately catches gray mold or that kind
Yes,
of
the
fungus.
strawberries will not keep it for longer days unless until they'll be packed in pouches and all.
Yes.
So this can be considerably kept for longer period. Maybe 2 weeks time is very good shelf life.
There are some varieties which have thick cuticles, so those type of berries they have better shelf life compared to there are some which are small when their size is very small and then cuticle is also not thick. So those ones may be they may have less, but still I think definitely more than a week.
Sir, this blueberry can be grown in Himachal Pradesh and Jammu and Kashmir, where are temperate regions.
Yes, yes. Temperature is low temperatures is a requirement because they are cold living plants. So chilling hour must be. Otherwise, you don't get that much fruit and yield. Your plant may well, but you may not get that fruit. And also, you cannot have the same variety. So if you want more fruit, you need to have different kind of varieties planted nearby. So that also increases the chances of getting food because they are self-pollination doesn't yield good crop, good fruits. So you need a different crop pollen, so that will help you to get large sized berries. So whenever they're planting, so it must be like maybe alternate row or that kind of planting you have to use, and then one variety, the other. So you can do that in alternate.
So we have to maintain isolation.
Not isolation, I am saying, but mix of the different cultivars. Same cultivar, you will not get
Okay.
better yield, more yield.
What is the pest and disease problem? What are the important pest and diseases in blueberries?
In blueberries, you don't get most the pest, but like just what was that called? Yeah, I don't remember that, but it doesn't have much of the pest problem.
Pest problem. Okay.
So I have one question.
Yes,
Madam,
ma'am.
please.
Actually, in this heat tolerance or heat stress studies, have you studied the nutritional value of the fruit? Because it is meant for the nutrition and medicinal value and health benefit is there.
No, we did not study the health, I mean the nutrition or what kind of components, because we, yeah, we know that the northern species, they have better composition related to like anthocyanins or other like a quercetin. These kind of flavonoids, they are better in northern species. So we know that. And then when we look at the composition of the southern or the berries which are grown in warmer temperatures, so that if you taste them, you will definitely understand that they are not good. But only reason why they are important because they can tolerate temperatures. So unless you do the introgression and then you do selections, so at that place you definitely need to, and okay, what is the, how much is because the once you, even if you do hybridizations, and then that linkage drag will be there, it will be compromised, the quality will be compromised. So definitely do those hybrids, you need to check for the composition and everything. But currently our object is only this, so we did not do that.
Okay, thank you.
Thank you, madam. Anybody is there? What is the price of 1 kg blueberries, sir, in US and India, I think?
Here, like in one box, probably like 200 gram or 300 gram, it costs like $2 to $3.
Okay.
Yeah, it is expensive, literally expensive. Yeah.
Uh-huh. Then other fruits.
Yes, compared to like if you compare with apple or those kind of, but definitely all berries are costly compared to other fruits. Yeah.
Okay.
Strawberries, blueberries, they have similar cost here in the US.
Okay.
Mhm.
Okay, thank you, sir. I think midnight at our place. So if nobody is there, I invite our registrar, Dr. V. Srinivasalis, to say a vote of thanks.
Thank you, Dr. Dhananjay Rao garu, the Director, Industrial and International Programs, for organizing the 21st Global Connect series by inviting Dr. Krishnanand P. Kulkarni garu, the Postdoctoral Research Associate from Department of Agriculture and Natural Resources, Delaware State University, USA. It's a very nice presentation made by Dr. Krishnanand on the displaying the genetic factors regulating heat stress tolerance in blueberries. I think today's speaker has a very good academic record also, because he is a product of IARI. That is one thing we should all appreciate him. And also he has worked on various crops like rice, chickpea, strawberries, and blueberries. And the present work is also very interesting because that crop is highly sensitive to heat. So there is every need to develop heat stress tolerant varieties in blueberries. I think the study on developing heat stress tolerant types in blueberries is of, the present day. It's very important in the context of America because it is mostly a cooler country. And the presentation is very good, excellent, with a lot of information on the genetics of this heat tolerance. We are very much thankful to Dr. Krishnanand Kulakarni garu for your nice presentation. I think the crop is new to us because we have been listening to this crop for several years. Strawberry, blueberries, and blackberries. But today, after your presentation, we came to know that this is very important, temperate fruit crop that has a lot to say in coming years because of its high health benefits. So your work will be of immense use, particularly the farmers of United States of America. As you said that you have already developed some, the 266 plants for heat stress tolerance types. So we hope that you'll come out with some good results, and that will be of very useful to the blackberry growers in your country. And we also request you one thing from our side. Every time our Honorable Vice Chancellor will be mentioning us whenever we speak of new varieties that are being grown in other countries. Is there any possibility to get those crops and introduce here in India? So this is one crop, I think, where we can introduce this crop in. Already some farmers are growing this crop in northern part of India, but there is every possibility to grow in southern parts of India also, provided that they can be given some suitable, regulated conditions. I think we request your help. If there is any, I think our Director Industrial International Program, Dr. Dhananjara, will also speak to you later. If there is any possibility of exchange of material, after getting some sort of MOU with Delaware State University, if there is any possibility, kindly help us to introduce some of the best varieties that can be suitable to our country. And thank you for your excellent presentation. It will really enrich our knowledge on blueberries, particularly on genetic aspects of the crop with regard to breeding varieties for heat stress tolerance. On behalf of Honorable Vice Chancellor and Director Industrial and International Programs and all the university officers and all the associate deans of horticultural colleges, principals of horticultural polytechnic, and also the faculty and students of Dr. YSR Horticulture University. We thank you all. Thank you for your nice presentation and sparing your valuable time for giving very excellent information on blueberries. Thank you very much.
Thank you, sir. Thank you.
Thank
It was a
you. One
pleasure.
more thing, sir, I want to ask Dr. Kulkarni, sir. Our Honorable Vice Chancellor is very much interested to have a signing MOU with our university. And all, not only our university, all Global Connect, this is the 21st Global Connect series. We have done with the 21 universities at international level. We want to have MOUs and to for exchange of students and research work. Any possibilities there, sir? We'll go ahead.
Yeah, sure, sir. We can talk about that maybe with our PI also, Dr. Kalpalata Milmai, and then we can talk what are the possibilities. But definitely there are possibilities.
Thank you. Thank you. I will send all the details and a letter. I will address a letter also, and definitely we'll have good relations and by signing MOU. And thank you very much for accepting our invitation to today as a guest speaker to present on blueberries. Thank you very much, sir.
Thank you, sir. Have a good day. Have a great day. Thank you.
Thank you, Dr. Krishna.
Thank you.
Thank you, sir. Thank you all university officers and all scientists and teachers, students. Now the program is closed. Thank you.
Thank you, Dr. Dhananjay.
Thank you, sir.
Thank you
Thank
for
you.
nicely organizing the program.
Okay, thank you, sir.
అప్పుడే అవ్వదు. ఎవరో ఉన్నారు హెచ్ఆర్ఎస్
Okay,